Okay, so as a bit of a reminder as to what we did, yeah. So the idea, remember, of this particular investigation is we're looking at whether different substances inhibit the growth of bacteria or microbes, yeah. Now keep in mind, what we're seeing isn't necessarily looking for whether it kills bacteria. We're just looking at whether it inhibits the growth. That's the key to it. Okay. So in your write up, when you talk in the discussion and things, you want to talk more in terms of, is it stopping the growth of the particular bacteria, not is it if it's killing the bacteria around what we've looked at. Okay. So what we did, Brendan, for your sake, is we looked at different antimicrobial agents. So they had to choose some different solutions to investigate. Yeah. So Owen, what solutions did you look at? Do you remember? Yep. So we looked at the antibacterial hand gel that's around the school, and you had a disinfectant that you looked at. Yeah. And oh, the dishwashing detergent. Yeah. So the dishwashing detergent that's found around the class. Yeah. This citrus detergent stuff. Okay. So they're the three that you chose. And did you just do all of yours at one concentration? Three concentrations? Okay, cool. So you did three solutions at three different concentrations. Cool. Yeah. Was it so the antibacterial gel, the detergent, and the disinfectant? Yeah. Did you do Listerine? No, somebody else did Listerine. We'll have a look at it in a second. Anyway, so you chose a range. Franco, do you remember what you chose? Perfume, that really crazy, like 70s antiseptic stuff. And a create. Oh, you used the Savlon, didn't you? Yep. Okay. So we chose three different solutions. And what we did was we took those solutions, yeah, and we took an agar plate and we used some E. coli bacteria and we spread out the E. coli bacteria on the entire plate, yeah, over the whole thing. All right. And then we took some discs that were soaked in our different solutions, placed them onto the agar plates, yeah. Oh, the mouth fresh, yeah, like the Listerine type stuff, the mouthwash. Yes, yeah, the mouthwash stuff. Okay, cool. Yep, and then we put them discs of those filter paper, which was soaked in our solutions, place them onto the agar plate. Okay, and the idea is that okay, so this is what it is. So they chose different chemicals, soaked them in some filter paper, placed them onto our agar plate, and we wanted to figure out whether this particular chemical inhibits bacterial growth. Yeah. Okay. And we said in order to measure whether there was inhibition of growth, we measure what we call a zone of inhibition. Okay. And a zone of inhibition is a way to give us an indication of how well it inhibits the growth of bacteria. All right. And so what we do, we use that as a measure. So we actually measure, you could measure the diameter, which is distance from side to side, the entire thing. You can calculate the area if you wanted to. So remember, the area of a circle is just pi r squared. So we could calculate the area. Or you can just use the radius. It's up to you. Yeah. At the end of the day, we're just comparing how large the zone of inhibition is compared to the other, I guess, the other types that we looked at. Yeah. Okay. So the greater or the larger the area, the more effective that compound or chemical is at inhibiting the growth of bacteria. That's the key to all of this. Okay. All right. So that's what you guys did. Now, because you all sort of did slightly different methods, because you looked at different things and different stuff. So part of the investigation was for you guys to write up your own method. Okay. Now I've given you a sheet that outlines sort of the basic background method. Yeah. And you can use this as a bit of a starting point. Okay. So it's got some of the materials that you use. So we used E. coli bacteria. Yeah. Okay. We used some nutrient agar plates. You guys used a hole punch and some filter paper. We didn't use the Bunsen burner in the end because we didn't have gas, so we just left that out. You did use a spreader. So the spreader is the orange thing that you use to spread the bacteria around with. Okay. You have your different test solutions, so you got to obviously list those that were appropriate to you. We used some sticky tape and we used some marking pens. Now, was there anything else that we used that I'm forgetting that's not on there? Tweezers, use some tweezers. Can't think of anything else off the top of my head. If we do remember, we'll list it, but I don't think so. Oh, we had the beakers as well. So we used some beakers to put the solutions in. Yeah. So you might want to add those two to the materials. Oh, sorry, over here. Forceps, which are tweezers. You did use some distilled water for the control. And you also have, obviously, the incubator, which is where we put the plates in. Yeah. So that gives you a bit of a basic outline of the different materials that we used. Okay. It's also got a method here. Now, look, it's probably, you could use this as a starting point, but yours will be slightly different. Okay. So, for example, 2 to 3 drops of bacterial culture in each of the plates. You did that. Yes. Okay. You didn't necessarily dip the spreader in methylated spirits or put it into the Bunsen burner because we didn't have the gas was turned off. Yeah, you did spread the bacteria evenly. You punched a number of discs of the filter paper. You soaked them, and you put the discs in the agar plate, looking something like this. Repeated for the other 3 plates. Incubated. Yes, great. So probably 95% of this is relevant to you. Some of it won't be. You can word it slightly differently. Okay. You can use that as a starting point, though. Okay. So let's have a look at the actual write-up and what are the different sections that we need, okay, and what you think you're going to put into each of these sections, okay. So it's given you a bit of background. So you've designed your experiment to figure out whether solutions can limit the growth of E. coli bacteria. That's what you're doing, okay. So let's think about what some of these sections are. So what should the title be for this particular investigation? Or what does a title generally need? And what do you think a potential title might be? So we talked about the differences between titles and aims before, okay? So inhibiting bacteria, yeah. Yeah, potentially. Any other ideas? So remember, with the title, don't overthink it too much, right? Generally, the title, you can pretty much use the title of the investigation, okay? So, well, I've pretty much given you the title, yeah? So it's essentially what you were saying. I would just use inhibition of bacterial growth. That's fine as a title, okay? It's just to give us some context as to what we're going to use, okay? So here, I've pretty much given you the title here. Probably don't need to put practical investigation part in, but your title might be something like, investigating different solutions on the inhibition of bacterial growth or something like that. Yeah, okay. How will that differ to the aim, though? So the title is pretty generic. Everyone can use the same title. That's fine. What about the aim? How will the aim differ, or how will that be different from the title? What is the aim going to be? Yeah. No, so the aim is more in terms of like we're literally stating what we're doing, right? And you're about 90% there. I like the idea of testing the effectiveness of something on something else. Yeah. Remember, we always generally want like what are we doing? Well, we're going to compare, right? So we're comparing generally. So we're going to look at, so we're going to compare the effectiveness of, and that's where you could list your specific solutions. So for you, it would be to compare the effectiveness of antibacterial hand gel, washing detergent, antiseptic, and mouthwash on the inhibition of growth of E. coli bacteria. Yeah, so it's quite specific. It's what are you specifically testing? So you're comparing the inhibition of growth. What are you comparing the inhibition of growth on? Well, of E. coli bacteria, and what are the solutions that you're comparing? Yeah, so you have to be really specific. Testing the effectiveness of these different solutions on the inhibition of growth of E. coli bacteria. That's what you are doing specifically, yeah? Nice and specific. So yours is going to be different to Franco's. So Franco's might be test the effectiveness of, antiseptic, Savlon, and, I can't remember, and perfume on the inhibition of growth of E. coli. Yeah, so nice and specific. What are you testing? Okay. Now, keep in mind, that's the other point I want to make now, and I'll repeat it later, is that all you are testing is whether it inhibits the growth of E. coli. That is it. You're not testing whether it inhibits the growth of bacteria. You're not testing whether it inhibits the growth of microbes. You're specifically looking at E. coli. Yeah. Now, if we were going to take this further, we'd do other bacteria, and we'd look at different things. In this case, all we're focusing on is E. coli. That's the only statement we can make. Okay. Variables. What's our independent variable going to be? Sorry? Distilled water, yeah, will be one. What else? No. Franco, what do you think the independent might be here? Yeah, that's what, remember, the independent is what you're choosing, what you have control over. So it's the solutions that you have chosen is the independent variables. Yeah. So this would be, you could write something like, yep, the solutions that we are testing, e.g., and then you would write down your specific solutions because this is what you have chosen to test. Yeah. Okay. So what would be the dependent variable here? What's the measured variable? No, so we're testing the effect that the solutions have on inhibiting the growth. The dependent is what are we measuring? How are we giving an indication of how well it inhibited? What are we actually measuring in this investigation? What did we call it? Started with a Z, the zone of inhibition. Yeah, cool. Yeah. So the dependent variable will be now it depends on what you're going to use. It might be like the diameter of the zone of inhibition or the area of the zone of inhibition. Whatever you want to measure, that would be the dependent. That's your measured variable. Yeah. Okay. And then the controlled variables are everything else. So they're things like, time in the incubator, temperature in the incubator, size of filter paper, nutrient agar used. All these things will be controlled. So everything else that we did has to be controlled variables. Okay. All right. Write a bit of an hypothesis. Okay. So this is what you predict will happen based on your prior knowledge. Yeah. Okay. Now, I want you to write this before you look at the results because it's really easy to fudge it later. I want you to give your honest opinion as to what you think will happen. So before you go up and get your results, I want you just to write down somewhere. You can type it out. I don't care what you think your hypothesis will be. Yeah? Okay? Now, you can use an if statement and then a then statement. So we could say, if... or you could say like if the, for yours it might be if the mouthwash is used, then the zone of inhibition will be 0. There won't be a zone of inhibition. However, if we, or you could say, the mouthwash will have the largest zone of inhibition, whereas the antibacterial gel will have a smaller zone of inhibition, but the detergent will have a 0 zone of inhibition or something. What are your thoughts? What do you think? And remember, always relating the independent to the dependent variable. Yeah? So you're relating what you think the solutions will be and what the zone of inhibition will be dependent on that solution that you're using. Okay? So I want you to write that up before you go and get your results. All right? Now do that in a second. We'll just get through this and then you can get onto it and have a go at it. Control group. What's the control group in this particular experiment? No. Brendan, have you heard of a control before when we do investigations? Maybe. So what's your idea? Why would we use a control group, and what do you think it might be in general?
Use some levels and what's produced on the experiment.
Not so much to do with sort of the labeling and all that stuff. Franco, you want to help us out? Yeah, you're on the right track. It will show us if there has been an error in the investigation. Got to remember, the idea of the control is to show that the independent variable is what is having an effect on the dependent. Yeah. So what we want to show is through the control is that if there is a zone of inhibition, that it's the solutions causing the zone of inhibition and not just something else weird that's going on. Okay. So in this case, we're using, what did we use as our control? What solution did we use for the control group? Distilled water, because we know that if we use distilled water, it should have no effect on the E. coli. Yeah. Okay. So in this idea, we want to show that the solutions are causing the zone of inhibition. Therefore, we use distilled water because we would expect E. coli to grow or to have 0 zone of inhibition where there is distilled water. Yeah. So if you get yours back and the control group shows that there's no growth, there's something wrong with your experiment. Yeah, there's an error there. Yeah. If you get it back and there is a zone of inhibition around your control group, something's gone wrong. So it's to show, one, that the E. coli will grow, and to two, that the zone of inhibition will only be present if there is some substance that will inhibit the growth. That's the key to it. Yeah. Okay. You will need to have some background related to the experiment. So what sort of background do you think you might include in this particular investigation? What would you include, do you reckon? Yeah, great. It's a good point. Yeah, so we would talk about maybe E. coli, what it is. Yeah, the fact that it's a bacteria. It's pretty common. It's found in a lot of places. This particular type is fairly harmless. But yeah, the fact, yeah. So what is E. coli? Yep. What else do you think? I think you probably want to include something around, the types of antimicrobial agents that you're using. Yeah. So maybe like what they're labeled as potentially. So you might be able, you might want to talk about some of the differences between like a disinfectant and an antiseptic and so forth, because I think you're both using different solutions. So you might just maybe want to touch on, what is an antimicrobial agent, and maybe what are some differences or types of antimicrobial agents, maybe. I think you're probably going to have to talk something around what is a zone of inhibition. Yeah, so what do we mean and why are we using that as a measure? Okay. So I think if you talk about what E. coli is, what are antimicrobial agents and what are some of the types that we are testing and what the zone of inhibition is. I think if you cover those three things, you should be all right in terms of background. Okay. Now, materials, you should be able to just take pretty much from this procedure list here. Okay. Whoopsie. So you should be able to, take out pretty much the materials from here. The method, you should be able to take 90% from that procedure list. You'll just have to change it a little bit yourselves. Okay. Now, results, what do you think you're going to include in the results here? What sort of things? What do you think you'll need in the results section? For me, I think a photo is a good idea. So just take some photos of your plates, chuck them on. Yeah, it's easy, visual. We can see straight away. All right, you're going to have a table of results. All right, so you'll need to show a table. Now remember, what are we measuring? We're measuring the zone of inhibition. Okay. So you need, these are the different solutions maybe. Here's the trials that we did. Here's the average, something like that. Yeah, you have to have the table of your entire results with some nice clear labels. What do we got? And you have to include any averages that you have across multiple tests of that one type. Yeah. And then you have to have a written description. Yeah. So remember we talked about in the results, just purely what do you see? Not the what yet. It's just, not the why. It's the what here. Yeah. So what are you seeing? Yeah. What are the results telling us? So it might just be, as can be seen from the results, the mouthwash showed no zone of inhibition, whereas the citrus detergent had a zone of inhibition of, 0.2 centimeters or whatever. Okay. However, we can also see that at different concentrations, we saw different zones of inhibition or something. And here's the trend that we saw or something like that. Yeah. So just telling us specifically what are we seeing based on the results. Then there's a discussion. So looking at 4 dot points. Okay. You don't really need to have this first dot point too much. If you've already given a good description of the results, probably don't bother with the summarizing the results. More this one, analyzing them and what they mean. Why do you think we're seeing what we're seeing? Okay. Now again, think about, in your hypothesis, you should have an idea of what you think will happen. Did you see what you thought you saw? Why or why not? What were the results? Explain what they mean. Yeah. So for me, if we look at this one, it's literally labeled antibacterial hand gel. So based on that claim, I would say I would expect this to have an effect. Yeah. And if it doesn't, there might be other reasons for that. Okay. Now, if it's not what you expected, try and come up with some reasons or why maybe some reasons why they didn't fit the pattern potentially. Now keep in mind there are reasons potentially why this may happen. Okay, so even though it's an antibacterial gel, will it work? Will it not? You expect it to. Maybe at different concentrations it might work better. Maybe it might just be too thick that it didn't leach out from the filter paper very easily. So you've got to remember that we're assuming that the solution is coming out from the filter paper to inhibit the growth. So part of it might be that this is just too thick and it hasn't had time to leach out. Maybe if it didn't work. I'm not sure. I'm just trying to think of some ideas as to maybe why that might have been. Yeah. Okay. So thinking about what the results, what they mean, were there any inconsistencies, potential errors maybe, and then what were some possible improvements? So what would you do next? What would be the next step? So for me, this is a fairly limited investigation. We're only testing one bacteria, yeah? So if we were going to test these solutions for real, I'd probably do multiple tests with different bacteria or different, maybe some fungi and maybe some different other types of bacteria. to give us a better indication of whether it's going to work on a range of bacteria. Because E. coli, you know, it looks a fairly common bacteria, but it's not going to be found everywhere. There's a range of other types of bacteria, so maybe that would be one improvement. And think about what some other improvements might be. Yeah. And that's it. There's a lot there to think about. All right. There's a fair bit to sort of write up. Any specific questions that you have at this particular stage? No? All right. If you don't have any questions, before you go get the results, write up a bit of an hypothesis as to what you expected. Grab some gloves when you're touching the plates. Now, keep in mind, theoretically, we're not actually meant to open the plates, yeah? So try and keep them closed and just measure it. You should be able to get a good measure from the back, yeah? Okay, so leave them sort of upside down, and you should be able to measure if there is any zone of inhibition from the back of it, yeah? Now, if you need rulers, I got rulers here. And Brandon will come and have a chat to you about what we did and what I'm going to get you to do going forward. It's just a health safety thing. Even though it's E. coli, we still can't guarantee it's safe. Well, we can, but we can't. I know, well, we weren't actually meant to. I didn't realise we couldn't, but I've been told by the lab manager we're not allowed to, so we shouldn't have opened those, but we did.